Today I'm going to be talking about functionalized polymeric electrospun nanoscaffolds for bone regeneration and tissue healing. I'm from University of California, Merced. Before I begin into my topic today, I just want to give you a little bit of information about University of California, Merced, and the foundry where we pursue these research projects. So University of California, Merced is the newest campus, the 10th campus in the UC-wide system. As you probably may be aware of other UC campuses like famous schools, UC Berkeley, UCLA, UC San Francisco. Likewise, they have 9 other campuses, and UC Merced was the last and the most recently established, about 6 years back established campus. And they have core research institutes within the campus, and I'm an associate professor and directing these core research institutes within the campus. So stem cell and nanofabrication instrumentation foundry is one of those core research units, and I oversee those units where we pursue research in various topics all the way from biology to nanotechnology to electrical engineering, optics, all different types of topics, because we have different equipments that can be used for different applications. My particular area of research is into nanobio, and we have 4 units in our foundry, mainly imaging unit, where we have all microscopes, scanning electron microscopes, TEM, confocal microscopes, and other reflected scopes. We have a flow cytometry cell sorting facility for mainly stem cell applications. And we have clean rooms like class 100 and class 1000 clean rooms where we do nanofabrication work, and we also have cell culture and tissue culture facilities. So my research projects are within all these 4 units, and today I'm going to be focusing on one project that kind of gets into all the different subunits of our institute. So I'm going to be discussing about the fabrication part of nanofibers, imaging part of nanofibers, and some analytical techniques that we use to analyze the composition of fibers, and finally the application of these scaffolds. So the objective of our work today is to fabricate nanoscale fibers and functionalize them for biomedical applications. The size and the structure of our fibers that we are expecting is within 500 to 700 nanometers range, and the choice of materials we had in a list that we selected. We first ranked as PLGA because PLGA is polylactic glycolic acid, very widely used, biocompatible, biodegradable polymer. It's FDA approved, so you can use this polymer for not only for ex vivo but also in vivo applications, and also for human trials, which we haven't done yet in US, but we intend to do that in the near future. So one of the objectives is to generate these functionalized PLGA fibers with that 700 nanometers range, and then we want to functionalize these fibers with demineralized bone matrix. So it's a bone powder that we put into the fibers along with other biomolecules that you will see in the next few slides what we add in there, and we want to try using these fibers for seeding different types of cells. So we use mesenchymal stem cells, we use cancer cells, we use different types of cells that we put on these fibers and put in the incubator and see if the cells really like these fibers as a platform. And then we study the elongation and the cell differentiation on top of these fibers, and then we also compare if the fibers are 2-dimensional versus 3-dimensional, if the fibers are random versus aligned, and then we study the comparison between the elongation and differentiation of those different types of cells, so we can study what type of fibers are actually more attractive for these cell units. So we use electrospinning technique to create these nanofibers. It's a very well-known and widely used technique for creating nanofibers. What you're seeing here is basically a high voltage power supply that we use. The positive bias of this high voltage power supply is going to a syringe. The syringe is which is full of a polymer dissolved in solvent, and there's a needle at the bottom of the syringe, which is metal needle, which is connected to the positive bias of the power supply, and the negative of the voltage power supply goes to a metallic ground. It could be simple aluminum foil or any metal that you would want to use, or silicon wafer. It depends upon what type of ground you want to use. And then we control this flow of the polymer that comes out of the syringe using the automated syringe pump. So with that control, we know exactly what flow rate we will be using to send the polymer liquid out there. Now what happens and how do we get nanofibers with this stuff, with this system here? So when you apply a high voltage power to a polymeric liquid solution that is flowing from the needle on the ground plate, the resulting electric field that comes out, it actually surpasses the surface tension, and as a result of that you start getting a whipping action for the liquid. So liquid starts getting into whipping action, and that sits on the surface of the ground plate here. When the solvent evaporates, you end up getting non-woven mesh type of nanofibers. And these nanofibers are like, we don't have any control on the size of the nanofibers as long as you know exactly what concentration, what flow rate, what power supply you're using, or what distance you're using to create these nanofibers. So there are 4 important key factors that really affects the size of these nanofibers or the diameters of these nanofibers. First, the potential that you're using, that means how much voltage you're applying. Second, the flow rate that you're applying for the solution to come out of the syringe. Third, the concentration of the polymer that you're using, like how much concentration with respect to the volume you're using. And the fourth, which is the distance between this needle and the ground plate. If you play around with these parameters, you would be able to control the diameters of the fibers the way you want. And that's exactly what we do. So we had to go through a lot of permutations and combinations in order for us to get the desired diameter that we are looking for a particular application that we are working. So that's how we create these fibers. And again, how this process works. So fibers go through the syringe needle, and then we charge them. We make sure that we get whipping action. Sometimes what happens is when you apply potential, like for example 10 kilovolts, you think that fibers, the solution is charged, but visually, if you cannot see that whipping action, you know that you won't get any fibers. So what you end up getting is big blobs of that liquid, or flaky structures, or beaded fibers. So you want to make sure that you get real good whipping action. For that, we have high resolution camera that we use in perpendicular to the jet stream that comes down, and then we can make sure that we get the right whipping action. And then that's how we control the diameter of the fiber. So as soon as the applied potential surpasses the surface tension, you know that it starts going to whip and it's going to come down. If you start at that point, start changing the distance between the needle and the ground plate, you can see large diameter distribution of the fibers. So we have built a automated XY stage on which we keep our metal ground plate. And if we want to have fibers with the diameters ranging from, let's say, 200 nanometers to 800 nanometers, we have developed a data sheet where we know exactly what parameters would give us 200 nanometers range fibers versus the parameters will give us 800 nanometers range fibers. So we start with the parameters that will give us 200 nanometers fibers, start the process, and then we program our stage in such a way that the stage moves in either X or in Y direction. And then depending upon which area we want 200 nanometers fibers or 800 nanometers fibers, we end up getting the fibers without breaking the whole process. So it's the same batch, but we get consistent fibers with different diameters. Because what we have noticed before that, even though you use the same concentration on Monday versus same concentration on Tuesday, you always have some variation in parameters, little bit of micro variation in parameters. You would never get the same type of fibers. So we thought we'd probably use large volume batch, and without breaking the process, we'll continue making the fibers from 200 nanometers to 800 nanometers, and that batch gives us most consistent fibers. So that's the point that I wanted to emphasize here, because we had to go through a lot of problem-solving methods to understand why this happens, and then we realized that process variation mainly depends on batch change. So if you don't change your batch of fabrication, then you won't have any probably issues with distribution of diameters of the fibers. So these are some of the parameters that we use to create fibers. This is just a cartoon of what you saw before in the previous image. This is a very crude way of showing how we use our fiber setup. And now we have more sophisticated units, commercially available electrospinning fiber units that you can purchase, but this is something that you can build with a minimum cost. This is the power supply, rotor stand, automated syringe pump, and your syringe, which you can hang it over here. And then we use different parameters for this PLGA fibers. We use 10% concentrations. We started from 2% and we go all the way to 20%. And then we also study, which particular concentrations give us the desired parameters we are looking for. The approximate applied potential that we use is around 15 kilovolts, and we use the flow rate of 20 to 30 microliters per minute. There are different types of needle gauges you can also vary, which is probably kind of, you can see, the fifth factor that can also affect the diameter of the fibers. But we normally use about 20 gauge needle to create the 700 nanometers range fibers out of it. And we have changed needles all the way from 20 to 25 gauge to study how that affects the diameter of the fibers. So once the fibers are prepared, made, then we need to study. We need to analyze those fibers. So we use SEM, scanning electron microscopy. We use TEM. We use laser confocal microscopy to study these fibers. And then we also use different types of assays to study if the mineralized bone that we mix with the fibers can really functionalize the fibers, and what kind of mineralization contents we can expect out of it. So then, like I mentioned previously, we use different types of cells to seed them on the fibers and incubate them in incubators. So these are the parameters that we use, typically 5% of carbon dioxide incubation at 37 degrees C, and approximate time for incubation is around 12 to 14 days. Then we do biocompatibility evaluation to understand whether the cells are alive or dead. It's basically cell viability study using the live dead cell staining and assay process. We also do immunohistochemistry staining that includes TAPI, osteocalcin, and collagen X. And then finally we use confocal and laser fluorescent microscopy to see if we can get or achieve two-dimensional versus three-dimensional fibers, because our ultimate aim is to get three-dimensional fibers, scaffolds, and we want to study if we get different stacks of fibers where we can seed cells in different stacks, and that's why we use the laser confocal microscopy for. So we use these analytical techniques to study the fibers. Now these are some of the images of scanning electron microscopes, in which the left column is basically what you can see here is the non-woven meshy structure of the fibers. This is the magnified version of this image with a scale of 10 microns, and this is a single fiber which was further magnified. As you see that we were able to achieve around 36 nanometer size fiber. Our goal is, again, depends on what application we are using these fibers for. Right now we are using the fibers for 700 nanometers range, but we also have other projects where we need like tens of nanometers of range of fibers, where we use the parameters to create 36 or 20 nanometers range fibers. So the column in the middle, where what we did was we took the silicon wafer and we patterned this gold serpentine structure because we wanted to study if we can use electrically conducting polymer to create nanofibers with the same parameters, and if we can study the electrical conductivity of those nanofibers across and see what kind of conductivity you get. So that was a different project, but I thought I should put these images here to show you that how we measure the electrical conductivity of the fibers. So basically we put the single fiber across the two metal pads and try measuring resistance across and see what kind of conductivity you can expect of that. And then the third column here, where you see different types of aligned fibers. Now these fibers are not aligned. These are like non-woven meshy fibers, but these fibers are aligned. And if you have aligned fibers, you can do quantitative analysis with aligned fibers, mainly if you want to use it for biosensing application or other applications where you need quantitative analysis. You know exactly how many fibers are. You can quantify what kind of enzyme or what kind of drug molecule or what kind of biomolecule you will be incorporating to these fibers, and then you can use it for sensing application or other applications. But in this case, you don't really know how many fibers are here. There's a large size distribution here. With aligned fiber structures, you can have better control for the applications that you're interested in. Now the question is, 2 minutes? Okay, so I got to go really fast on that. So this is the process, how we get the fibers. This is the whipping action that you can actually visually see without using any high resolution camera. But if you have high resolution camera, you can see this action very clearly, and that tells you that you're definitely going to get nanofibers out of this unit. So these are some of the biocompatibility tests we did. The red color shows dead cells versus green color shows the live cells, and then we did comparison between PLGA film versus PLGA fibers that we fabricated, and we saw that there's almost none dead cells in the PLGA fibers. So that means the cells will like that structure and the material. We also did some fluorescent images using collagen X and osteocalcin, and also some confocal images using those stains to study the cell properties and elongation and differentiation models. We did some immunohistochemistry data. Here you see 5% PLGA fibers, 10% PLGA fibers, and 10% PLGA film on the left, and the same composition on the right with different types of staining, collagen X and osteocalcin expressions. So the idea was to study different stains and see whether if we use osteocalcin versus collagen X, what difference we get, and we hardly saw any significant difference between the studies. So we thought we can use both type of staining procedures for that. Again, with these images, I want to show you how non-woven fibers could be made, aligned fibers, and then we also stained those fibers using a dye and got the confocal image of that showing the fibers could be aligned, and then we seed cells on those dyed fibers, so we can actually see cells seeding on the fibers. These are the images of the mineralization assay, what we did. So there's a petri dish with the PLGA solution in, and we dyed that. We put the mineral DBM into that, and then you can see different concentration, 10% fibers, 10% aligned fibers, and 10% PLGA films. And what we saw is the most darkest color of this concentration that tells us that the maximum mineralization happens in 10% aligned fibers, which is what we were expecting. I'm going to really go fast and just summarize these points, which is what we found on notice was if we compare PLGA film versus PLGA random fibers versus PLGA aligned fibers, the most promising results that we got out of PLGA aligned fibers, where we were able to seed human mesenchymal, adipose-derived mesenchymal stem cells, and we were able to study differentiation consistently as compared to putting on PLGA film or PLGA non-aligned fibers. There are multiple applications of nanofibers and endless opportunities where you can use nanofibers for. I just wanted to show you this slide and tell you, what areas like nanosensors, cosmetics, military applications, optics, in different fields you can use nanofibers for. Besides the scaffolding applications, currently my students are also working on using these fibers for drug delivery system and also for creating the nano bandages for wound healing applications. So we are using PLGA fibers, we are using conducting polymer fibers, and we are using composites of fibers for all the way from optoelectronics to biomedical applications, and we have a wide range of instrumentation that we use for studying these structures on campus. So with that, I think I'll wrap it up because I'm running out of time, but I really thank you for listening to my talk, and I would be happy if you have any questions to answer.